if we could see inside the cell exactly how cancer therapies break DNA and measure it with X-ray-like precision? That's the promise of Stride, a technology we created to solve a critical challenge: how to directly detect and measure DNA damage with precision right where it happens. Before Stride, researchers trying to see individual DNA lesions under a microscope saw nothing. No tool could label single DNA breaks. As scientists studying cancer biology, we faced the same challenge and decided to develop our own solution. That's how Stride technology was born. With Stride, instead of seeing nothing, we saw this. The blue ovals are cell nuclei. The red dots, called foci, represent individual DNA breaks formed in response to drug treatment labeled with Stride. Just as X-rays transformed bone diagnostics, Stride reveals how therapies affect DNA inside cells, answering key questions: how many breaks, what type, where. It works on research samples and patient samples. It detects even a single DNA lesion. By using Stride, pharma companies can develop safer, more effective, more precise therapies and bring personalized treatments to patients faster. Curious how Stride could support your work? Let's connect.